Okay, so the last step we're going to do in lab this week is to set up the polymerase chain reaction. So here's a quick um, little lesson on what PCR chain reaction is. And it is truly the foundation for modern biology. So in a second I'll get to exactly what it is and how it works. Now I want to talk to you a little bit about what it's used for. PCR forms the foundation of all of molecular biology and cell biology. Really, I can't imagine a molecular biology lab that doesn't use PCR on a daily basis for any part of their projects. Anytime you hear of splicing a gene into something or out of something, that's going to have to use, that often uses PCR. In terms of technology, we've been able to use it to create bucket loads of insulin by splicing the gene for insulin into E. coli and then making the E. coli make the insulin. We can use it, as we're using it here in our project, to identify what kind of species we have. We can use it to determine how related two species are. It spans the gamut between molecular and cell biology all the way out to ecology and evolution, and everywhere in between uses this technique. It's that foundational to modern biology. The Forest Service uses it. The Fish and Game Service uses it. It's ubiquitous. It's everywhere. As we go through this little video, I'll talk about what we're using it for. We've already talked about what we're using it for to a certain extent, but we'll get more detailed here. So fundamentally, PCR is a process for replicating DNA in a test tube. And the very first person who did this won a Nobel Prize for being able to do it. And since then, in refining that process, there have been probably close to 4 or 5 more Nobel Prizes awarded for different refinements of the process of replicating DNA in a test tube. And we've moved it from the macro scale, where we were using visible quantities of things, to the micro scale. As you'll see during lab, some of the quantities we use are barely visible to the naked eye. So unlike a normal organism or cell that replicates all of its DNA just once prior to reproducing, in the lab we use PCR to make billions of copies of just one small section of that DNA. For that, our project, we call that section of DNA the gene of interest. So now a word about our gene of interest. So usually we think of genes as coding for proteins. In other words, the product that's made from that gene is messenger RNA. That messenger RNA then finds a ribosome and is translated into a protein. That's usually how we think of it. But, excuse me, some genes code for ribosomal RNA, and our gene of interest is one of those. It codes for the 18S ribosomal RNA. If you remember, ribosomes are mostly composed of RNA. Not only is the structure RNA, but the enzymatic function of a ribosome, in other words, connecting two amino acids to make a protein, that is done by the ribosomal RNA. It's one of the few places where RNA is an enzyme, or an enzyme is made of RNA rather than a protein. We use this particular gene very commonly to distinguish between members of closely related species, because for a couple of reasons. Number one, every living cell has ribosomes. Why? Because ribosomes are what make proteins, and proteins are needed for every chemical reaction of life. So you can't have a cell, a functioning cell at least, without ribosomes. So these genes are ubiquitous. They come in different forms between prokaryotic and eukaryotic, but they all go back to come from the same original RNA that served as a ribosomal RNA. They all evolved from that RNA, so they're all related. That's one reason it's widely used. Secondly, this gene is highly conserved. That means it does not accumulate mutations very quickly in evolutionary time. In order for a molecular clock to be useful, it needs to accumulate mutations over time in a fairly regular way. Not to mean the mutations are regular, they're random, but you only get a certain number of mutations per million years. That makes for a good molecular clock. And because it's highly conserved, this gene that codes for the 18S ribosomal RNA is highly conserved and makes a good molecular clock. If it mutated very quickly or in some other fashion, or it never mutated, it would not make for a good molecular clock. By molecular clock, I mean a way of determining how long ago did these two species diverge in evolution. So let's take a quick look here at how DNA is replicated inside a cell naturally versus how it's replicated in a test tube, in PCR. So the first step in cellular DNA replication begins with helicase, an enzyme that requires ATP to create single-stranded DNA. It unwinds the DNA into what's called a replication bubble, so that a small region of DNA is separated into single strands that give the polymerase access to make the copies. In our case, we're going to use a lot of heat. We're going to raise it to very nearly boiling point. And that's enough to separate the DNA into single-stranded DNA, a process called denaturing, which is related to but somewhat different than denaturing proteins. The second step inside the cell is for an RNA primase to come in and lay down a little section of RNA to give DNA polymerase a starting point. Because DNA polymerase can't start de novo. It can't just go in and lay down the first nucleotide. It has to have something to attach that nucleotide to. So you put in a little RNA primer, then start elongating the DNA from that. That means ultimately that you have to go back in later, take out that RNA, replace it with DNA, and seal the nick. Kind of an involved process. Here, we're just going to use artificially made DNA primers. It gives us a couple advantages. First of all, we decide what the sequence of that primer is. And secondly, it's DNA, so we never have to go in and replace it. It just becomes part of the new molecule. The next step inside the cell is for DNA polymerase to actually do the elongation. And for most organisms, that DNA polymerase, for example, our DNA polymerase works best at a temperature of 37 degrees Celsius, our normal body temperature. We're going to use a special type of DNA polymerase that comes from an organism called Thermaticus aquaticus. This is an organism that evolved in the hot springs in Yellowstone National Park, where the water temperatures get very close to boiling, if not boiling itself. That means these organisms needed a protein, because this is an enzyme, it's a protein that won't denature under extraordinarily high temperatures. This part of the process was one of the steps that won a Nobel Prize. This allows us to do this inside a test tube in a lab functionally in a meaningful way, in a practical way. And I'll explain why that is later on. In regular cell, you need to worry about leading and lagging strands. In PCR, that's not going to be applicable because we're going to have completely separated. We're not dealing with a replication bubble, but two complete separate strands, single strands. Inside a nucleus is a very specialized environment. That cytoplasm inside the nucleus, or nucleoplasm, has a carefully regulated pH and ionic concentration and other attributes. We mimic those with an artificial pH/ionic buffer. The key to PCR is to avoid using as many enzymes as possible. In other words, use as few enzymes as possible. Here, helicase, RNA primase, DNA polymerase, add in the replacing of the RNA primer and the ligase, and you've got half a dozen different enzymes that the cell uses. In PCR, we've winnowed that down to one enzyme, the DNA polymerase, and we found a particularly hardy form of that polymerase that we can use. So let's look at the steps that PCR uses. The first step is to heat the DNA up to 94 degrees, which is going to denature or separate the DNA into single strands. Then we cool it down to 55 to 65 degrees, and that exact temperature depends on our exact artificial DNA primers. I believe our exact temperature is somewhere in the low 60s. At this temperature, the separate single-stranded DNA won't recombine. It won't go back to being double-stranded. You keep those single strands, but it will allow these short stretches of DNA to anneal or bind to their specific region of the DNA. So these primers have two functions. The first one is they're going to be the starting place for the DNA polymerase to elongate the new DNA strands, but they also specify exactly what part of this entire huge genome we want to copy. Then step 3, we heat it up to 72 degrees Celsius, which is the optimal temperature for Taq polymerase to elongate the 2 new strands of DNA. Step 4 is to go back to step number 1 and repeat. So it's going to wash, rinse, repeat, and we'll do that 30 to 34 times around this cycle. Each time around, you double the amount of DNA. So the copies of your gene double with each round of PCR. That's a geometric growth. So the first round gives you 2 copies, the second round 4, then 8, then 16, etc. And by the time you get to 34, you literally have billions of copies of your gene of interest. So the first step here is this double-stranded genomic DNA, heated up to 94 degrees, very nearly boiling point, which is going to separate them out into single-stranded DNA, denatures them. Cool that back down to about 60, and that allows for the annealing of our primers. We'll have a forward primer and a reverse primer in our reactions, and they are complementary to a specific site in the entire genome. There will be just this one site that this primer is complementary to the base sequence of the template DNA. And this primer will be complementary to this base sequence in this DNA. We'll talk a little bit more about that in lab down the road, not this week. So we've annealed our primers to our gene of interest. So they bracket our gene of interest, which falls in this region. Next step, heat to 72 degrees. That's the optimal temperature for DNA polymerase to elongate 2 new strands of DNA. And we're left with 2 new copies of our gene of interest. Now, this is what makes Taq polymerase so special, because the next step, you heat it up to 94 degrees again. Ordinarily, that would kill an ordinary polymerase. It would kill our polymerase. It would completely denature it, and so to go to the next round, we would have to add more of that polymerase. So each one of those 34 rounds of PCR that get done, you have to stop and add more polymerase. That's not particularly functional. It takes an extraordinarily large amount of time. It changes the volumes involved. It changes the concentration of your buffers. It gets very, very complicated. Having a polymerase that can withstand 34 degrees or 94 degrees without denaturing allows you just to repeat this process on end without having to stop. So you just keep repeating that 34 times, and you come out the end with billions of copies. And I would say that this reaction is going to take about an hour and a half to complete. Now that's going to be done outside of lab. You're going to set up the reactions, and I'll do the set the machine going, but it'll only take about an hour and a half to create billions of copies of our gene of interest, this particular region of the entire genome. So we're going to set them up immediately after you isolate your genomic DNA this week. So we're going to use pre-prepared reaction tubes. That comes with some stuff already in the tube. These are DNTPs, and the N is shorthand for adenosine, thymine, guanine, and cytosine. So all 4 of your bases, and they're DNTPs because they're deoxynucleotides to make DNA. And a 2x pH ionic buffer. There'll be 25 microliters volume in here already. We're going to add our template DNA, that's what we just isolated from the unknown algae, both the forward and reverse primers, the Taq polymerase, and some molecular biology water. The volume of all of this stuff will also add up to 25 microliters. So 25 and 25 equals 50. This 25 microliters dilutes this to a 1x working buffer. and that's what we'll be doing the very last, and then I'll take your tubes and set them up. Now, after watching this video, you can follow the link to Blackboard that gives you an animation of how that amplification of the number of copies of genes actually occurs.